Hello and welcome to this presentation. Platform name is Blackboard Research and Foundation, and I will be your presenter today. Today we will be discuss visual pathway and anatomic nervous system. Introduction. The visual pathway refers to the series of structures and processes involved in transmitting visual information from the eyes to the brain, where it is interpreted as the image we see. The pathway begins with the retina in the eye, which contain specialized cells called photoreceptors that convert light into electrical signals. From their signals travel through the optic nerve, which is a bundle of fibers that carries information from the retina to the brain. The optic nerve passes through the optic chiasma, where some fibers cross over to the opposite sides of the brain and others remain on the same sides. Next, the signal travel through the optic tract to various parts of the brain, including the lateral geniculate nucleus in the thalamus and the primary visual cortex in the occipital lobe, occipital, and these structures process to the information and contribute to our perception of the visual stimuli such as colors, shapes, and movements. Overall, the visual pathway is a complex system that allows us to see and interpret the world around us. The visual system is responsible for processing visual information and allowing us to perceive the world around us. The retina is a key component of the visual system located in the back of the eye that is responsible for converting light into the electrical signal that can be interpreted by the brain. So this slide I will discuss regional anatomy of the visual system, retinal structures. The retina has several layers of cells, each with a specific functions. The outermost layer of cells, called the photoreceptor layer, contain two types of cells, rods and cones. Rods are more sensitive to low level of light and are responsible for our ability to see in dimly light environment. Cone, on the other hand, are responsible for our ability to see color and fine details in bright light. The photoreceptor layer is the bipolar cell layer which receives signals from the rods and cones and passes them on the ganglionic cells. The ganglionic cells are the output cells of the retina and transmit visual information to the brain via the optic nerve. In addition, the visual system in the retina consists of the photoreceptor, bipolar cells and ganglionic cells. The photoreceptors are responsible for absorbing light, converting into the electrical signal and sending it to the bipolar cell. The bipolar cells in turn send the signal to the ganglionic cells, which then sends signals to the brain visual cortex. The retina also contains amacril cell which help to regulate the flow of information within the retina. Overall, the regional anatomy of the visual system, the retinal structure is complex and highly specialized, with each layer of cells playing a critical role in a processing and transmissions of visual information to the brain. Optic nerve and optic chiasma. The optic nerve is a cranial nerve that connects the eye to the brain. It is responsible for transmitting visual information from the retina to the brain, allowing us to see. Each eye has its own optic nerve, which exits the back of the eye and enters the brain through the optic canal. The optic chiasma is a structure located at the base of the brain where the optic nerve from both eyes intersect and partially cross over. At the optic chiasma, some of the nerve fibers from each optic nerve cross over to the opposite sides of the brain, while other contain on the same side. The crossing over is important because it allows the brain to receive visual information from both eyes, which is essential for depth perception and other aspects for vision. After the optic chiasma, the nerve fibers that originated from the same side of each eye joined together from optic tract. The optic tract then contain to the visual cortex in the back of the brain, where the visual information is processed and interpreted. Lateral geniculate body. The lateral geniculate body is an important structure in the visual system of the brain, and it is involved in processing visual information from the eye. In ophthalmology, the lateral geniculate body is of interest because it is the location where the optic nerve fiber from the eyes synapse with neurons that project to the primary visual cortex in the occipation lobe of the brain. This means that any damage to the lateral geniculate body, such as from stroke or a tumor, can result in visual field defect or other visual abnormalities. Visual field defect can manifest in different way depending on which part of the lateral geniculate body is affected. For example, damage to the upper part of the lateral geniculate body can result in visual field defect in the lower part of the visual field in both eyes. Similarly, damage to the right lateral geniculate body can result in visual field defect on the left side of the eyes. Ophthalmologists may evaluate the functions of the lateral geniculate body using specialized tests such as visual evoked potential or magnetic resonance image, that is called MRI. These tests can help to locate where is visual field defect are there in a lateral geniculate body. Optic radiation and projection to the primary visual cortex. The optic radiation is a bundle of axon that carries visual information from the lateral geniculate nucleus LGN of the thalamus to the primary visual cortex in the occipital lobe of the brain. The optic radiation can be divided into two main components. The optic radiation can be divided into two main components: the geniculocalcarine tract and the superior longitudinal fasciculus. The geniculocalcarine tract is the main path that carries visual information from the LGN to the V1, while the superior longitudinal fasciculus connected the V1 to the other visual process area in the brain. The projections from the optic radiation V1 organized in a retinotopic manner, meaning the night boring areas of the retina project and night boring areas of the V1. This origination allow to the brain to create a specially accurate representation of the visual world. Once visual information reach the V1, it undergoes further process by different types of neurons in different layer of the cortex. The process includes features extension such as age detection and orientation sensitively and integration of information from both eyes to create a unified visual perception. Overall, the optic radiation and projection to the primary visual cortex area essential component of the visual pathway, allowing us to perceive and interpret the visual world around us. Blood supply to the visual pathway. The visual pathway refers to the pathway that carries visual information from the eyes to the brain where it is processed and interpreted. The blood supply to the visual pathway is critical for the health and function of the visual system. There are several key arteries that supply blood to the visual pathway. Number 1, ophthalmic artery. This artery is the first branch of the internal carotid artery and supplied blood to the retina, optic nerve and other structures in the eye. Number 2, central retinal artery. This artery branches off from the ophthalmic artery and supplied blood to the retina. Third number, posterior ciliary artery. This artery also branches off from the ophthalmic artery and supply blood to the choroid, which is a layer of tissues in the eye that provide oxygen and nutrition to the retina. Number 4, arteriocordial artery. This artery arises from the internal carotid artery and supplied blood to the optic tract, lateral geniculate nucleus and other structures in the brain that are involved in visual processing. The blood supply to the visual path is circular for maintaining the function of the visual system. Any disruption of blood flows to these structures can result in visual impairment or blindness. Neural pathway for vision. The first order neural in the visual pathway is the bipolar cell of the retina. These cells receive visual information from the neural pathway cells of the retina, such as rods and cones. The bipolar cells then send the information to the ganglionic cells, which are second order neuron of the visual pathway. Second order neurons in the visual pathway are the ganglion cells of the retina. These called are responsible for transmitting the visual information from the photoreceptor to the lateral geniculate body of the thalamus. From there, the information is then sent to other part of the brain for further processing. The ganglionic cell of the retina contain axons that extend along the optic nerve and through the optic chiasma. The axons are responsible for conveying the visual information from the eye to the brain. The nasal fibers of the optic nerve originate from ganglion cells in the nasal half of the retina. After leaving the eye, these fibers passing through the optic chiasma and then enter the contralateral optic tract. This process is known as discussion, where fibers cross over to the opposite sides of the brain. Once in the optic tract, the fibers relay visual information to the visual cortex in the occipital lobe of brain. The temporal fibers of optic nerve originated from ganglionic cell in the temporal half of the ipsilateral retina and the contralateral retina. These fibers joins together at the optic disc and are redirect to the optic chiasma, where they cross to the opposite side before entering the optic tract. From the optic tract, the fibers travel to the lateral geniculate nucleus and then on to the visual cortex in the occipital lobe. Neural pathway for vision. Continue. The cell body of first order sensory neurons are located in the lateral geniculate nucleus, are also as the lateral geniculate body. Lateral geniculate body is the structure in the thalamus that is part of the primary visual pathway. It received information from the retina and sent it to the visual cortex. The cell body is these neurons are located in the nucleus, where they received and processed visual information from there. The neurons send visual signals to the visual cortex, where they are further processed. The axon from the optic radiation, also known as the geniculocrine tract, project into the primary visual cortex, which is located into the occipital lobe of the brain. These axons carry visual information from the eyes to the visual cortex, where it is passed and interpreted to form our perception of the visual world. The optic radiation is made up of axons that originated the lateral geniculate nucleus of the thalamus, which received input from the retina of the eye. The axon then passed through the posterior limbal of the internal capsule and terminate in the primary visual cortex, also known as V1 or the straight cortex. The visual cortex is organized in a way that allow from the processing of various aspects the visual information, such as color, motion, and spatial frequency in different regions of the cortex. Visual reflexes. The light reflex, also known as the pupillary reflex, is a reflex that controls the size of the pupil in response to change the light level. The reflex is controlled by the automatic nervous system and involve the construction of dilation of the pupil. When light travel change, specialized cells in the retina called photoreceptors send signals to the brain. The brain then sends signals to the muscles in the iris, which control the size of the pupil. In bright light, the muscles constrict the pupil, reducing the amount of the entering light. In dim light, the muscles dilated the pupil, allowing more light to enter the eye. The pupillary reflex is an important diagnostic tool used by healthcare professionals to assess the function of the nervous system. Abnormalities in the pupillary reflex can indicate a variety of neurological conditions, including damage to the brain system, optic nerve, or automatic nervous system. The pathway for the visual reflex. The pathway for the visual reflex that control the construction for this sphincter pupil in responsible to the light stimulus involving several structures in the brain and the eye. First one, light enter the eye and detected by the photoreceptor cells in the retina. Secondly, the information from the photoreceptor cell transmitted via the optic nerve to the lateral geniculate nucleus in the thalamus. Thirdly, from the lateral genicular nucleus, the information is transmitted to the primary visual cortex B1 in the occipital lobe of the brain. Fourthly, some of the information from B1 is transmitted to the protection area in the midbrain, which involve the controlling the pupil size. Fifthly, the protectal area sends signals to the Edegner Westphal nucleus, EWN short form is, in the midbrain, which contain the preganglionic parasympathetic nerves or neurons that control the sphincter pupil. Sixthly, the paraganglionic neurons from the EWNs synapse with the photoganglionic neurons in the ciliary ganglions. Seventh, postganglionic nerves project to the iris sphincter muscles, causing it to contract and decrease the size of the pupil. The pathway allows for a rapid and automatic response to change in light intensity, help to protect the retina from damage and improved visual acuity in bright environment. Pathway of direct and indirect reflex. The direct and indirect light reflex pathway in ophthalmology involves the stimulation of the retina by light, which then travel along the optic nerve to the fractal nucleus, which then sends signals to the EW, and from their signals are sent to the ciliary ganglion, which then sends signals to the ciliary muscles that control the size of the pupil. The indirect pathway, the signals travel directly from the retina to the EW and then to the ciliary ganglion. In the indirect pathway, the signal travel from the retina to the protectal nucleus, then to the EW and Edinger-Westphal nucleus, that is, and then to the ciliary ganglion. This slide I am showing you pupil construction as the circular muscle of iris construct, and pupillar dilate as a radial muscles of iris construct, that is sympathetic. And what is the normal pupil? Bright light is parasympathetic pupil construction, and dim light pupil is dilated, that is sympathetic. That is the view of pupil in different condition of light illumination. The automatic nerves to the eyes. The autonomic nervous system play an important role regulating the functions of the eyes. The autonomic nervous to the eyes, the two branches of the autonomic nervous systems is sympathetic and parasympathetic, have opposing effect on the pupil size and the shape of the lens. The sympathetic nervous system stimulate the dilator muscles in the iris, causing the pupil to dilate or enlarge. This response is important in low light condition to allow more light to enter the eye. The sympathetic nerve systems also increases the tone of the ciliary muscle, which change the shape of the lens to allow for clear distance vision. On the other hand, the parasympathetic nervous system stimulates the sphincter muscle in the iris, causing the pupil to contract or shrink. This response is important in bright light conditions to reduce the amount of light entering the eye. Parasympathetic nerve system also decrease the tone of the ciliary muscles, which allow this lens to become more rounded for near vision. Therefore, the autonomic nervous system play a critical role in regulating the functions of the eye, including the size of the pupil and the shape of the lens. Accommodation reflex. The accommodation reflex is a physiological response that occurs in the eye when it changes focus from an object at one distance to an object at a different distance. Especially, the accommodation reflex involves change in the shape of the lens in the eye, which allow the eye to focus on object at different distance. When an object is at distance, the ciliary muscle in the eye contract, causing the lens become more convex or around. They allowed the lens to refract light more effectively and focus onto the retina at the back of the eye. Conversely, when an object is closer to the eye, the ciliary muscle reflects, causing the lens to become flatter. They reduce the refractive power of the lens, allowing the eye to focus on the near object. The accommodation and reflex is a necessary process for clear vision at various distance, and it is controlled by the autonomic nervous system, which regulates involuntary body functions. Clinical correlation. Homeopia is a clinical term used to describe the loss of one half of the vertical visual field. It is usually caused by brain damage and can result in a loss of vision in the right or left half of the visual field. Symptoms can including impaired color vision, reduced contrast sensitivity, and difficult recognizing object in the affected field. Treatment options including glasses, contact lenses, eye exercise, and medication. In some cases, surgery may be needed to correct the underlying causes of the vision losses. Homonymous homeopia or hemianopia is a condition in which a person loses vision in the same halves of the vision field of each eye. It is usually caused by damage to the brain, such as a stroke and head trauma. Symptoms of homonymous hemianopia can include difficulty reading and recognizing face, difficulty finding object in the environment, the difficulty navigating in unfamiliar place. Treatment options for homonymous hemianopia vary depending on the cause and severity of the vision losses, but can including visual training, vision therapy, and medication, that is management of homonymous hemianopia. Heterogeneous homeopia is a visual field defect that affects both eye and result in a loss of vision on one side of the visual field in each eye. This means that the person with this condition cannot see object on the same side of their visual field in both eyes. Heterogeneous hemianopia can be caused by various factors such as the brain injuries, tumor, or stroke that damage the visual pathway in the brain that transmit visual information from the eyes to the brain. This severity of the visual field loss can vary depending on the location and extent of the damage in the brain. People with heterogeneous hemianopia may be experience difficulty with the activities that require them to perceive and respond to the object on the affected side to their visual field, such as driving or reading. However, they can learn to compensate of their visual impairment by using traits such as scanning their environment for more frequently or turning their head to the see object on the affected side. Visual field defect due to lesions in different parts of the visual pathway. Visual pathway defect occur due to lesions at different parts of the visual pathway. The visual pathway include the structures of the eye, optic nerve, optic chiasma, optic tract, lateral geniculate nucleus, optic radiation, and the visual cortex. Each of these structures has a specific role in processing visual information, and damage to any of them can result in different type of visual field defect. Different type of visual field defect. Here are some examples. First, lesion of the optic nerve. A lesion of the optic nerve can result in a complete loss of vision in the eye affected by the lesions. This is because of optic nerve carries visual information from the eye to the brain, and damage to the nerve can interrupt this communication. Number 2, lesion of the optic chiasma. The optic chiasma is the point where optic nerve from the both eye meet and cross over. Lesion of the optic chiasma can result in specific visual field defect depending on the location of the lesions. For example, a lesions on the bottom of the optic chiasma can result bitemporal hemianopia, where outer half of the both visual field is lost. Number 3 and 4 and 5, lesion of the optic tract. The optic tract carries visual information from the optic chiasma to the lateral geniculate nucleus in the thalamus. Lesion of the optic tract can result in specific visual field defect depending on the location on the lesions. For example, a lesions on the left optic tract can result in a right hemianopia, where the left half of the visual field is lost in both eyes. Number 6, lesion of the lateral geniculate nucleus. The lateral geniculate nucleus is a relay stations in the thalamus that processing visual information before sending it to the visual cortex. Lesion of the lateral geniculate nucleus can result in specific visual field defect depend on the location on the lesions. For example, a lesions in the upper part of the lateral geniculate nucleus can result in a lower contralateral quadrennial where a lower quarter of the visual field on the opposite side of lost. Another is lesion of visual cortex. The visual cortex is located in the occipital lobe of the brain and is responsible for processing visual information received from the thalamus. Lesions of the visual cortex can result in specific visual field defect depending on the location of the lesions. For example, a lesions in the right occipital lobes can result in the left homogeneous hominopia, where the right half of the visual field is lost in both eyes. In summary, visual field defect can occur due to lesions at different parts of the visual pathway, and the type and location of the defect depend on the location of lesions. Argyll-Robertson pupil. Argyll-Robertson pupil (ARP) is a medical condition characterized by small irregular pupils that do not react to change in light but construct during near vision. The condition is named after this Scottish ophthalmologist Argyll Robertson, whose first described in 1869. Argyll-Robertson pupils are most commonly associated with neurosyphilic at late stage complication of syphilic infection. However, they can also occur as a result of other neurological conditions, including multiple sclerosis, diabetes, and tumor affecting the midbrain. The diagnosis of ARP is made by an ophthalmologist who performs a detailed eye exam, including an assessment of the pupil response to light and near vision. Treatment typically involves addressing the underlying cause of the conditions, such as antibiotic for syphilis or other targeting therapeutic for specific neurological conditions. Thank you for watching my video.